Hello, and welcome to the Virtual Anesthesia Symposium of 2020. My name is Jina Park, and I am currently a high school senior at Paul Lawrence Dunbar High School and researching under the mentorship of Dr. Bjorn Bauer and Dr. Annika Hartz at the Sanders Brown Center on Aging. In this video, I'll be presenting to you my research project titled TNF-alpha in serum of glioblastoma mice. Now, before we dive right in, I'd like to go over a brief overview of the video. First, I'll introduce a bit of background information, as well as present the overall research question that drove this entire project. I'll then go over the procedure that allowed me to test this question, and then dive right into the results and conclusion that I was able to form from the experiment. I'd like to finish the video with a few acknowledgments and a brief look at my references, and I'll leave my contact information for anybody who has questions. So let's get right into it. So what exactly is TNF-alpha, and what does it mean when I say glioblastoma mice? Well, to understand this experiment in its entirety, we're going to have to dive into a little bit of background information, starting with something called glial cells. The nervous system within our bodies consists of two main types of cells, neurons and glial cells. While neurons have the important task of passing along signals from brain to neurons, glial cells play more of a supporting role. There are many types of glial cells, but the ones that we'll focus on for this project are the ones called astrocytes. And astrocytes are star-shaped glial cells located in the brain and the spinal cord. They have a variety of functions, like regulating blood flow in the brain and contributing to the formation of the blood-brain barrier, which is, by the way, something we'll touch on later in the video. Okay, well, but what does any of this have to do with glioblastoma? Well, glioblastoma, or glioblastoma multiforme, is a grade 4 astrocytoma, which are brain tumors that originate from those astrocyte cells. And grade 4 means that glioblastoma is one of the most aggressive and invasive forms of brain cancer, with a tumor that rapidly grows and spreads into nearby brain tissue. Symptoms start off slow. Patients may experience headaches, nausea, and dizziness at first, but as the cancer worsens, symptoms spiral into things like memory problems, cognitive impairments, and seizures. The median life expectancy for patients with GBM ranges from only 15 to 16 months, which really isn't a whole lot, and there is no definite cure. And although surgery is used to remove the primary tumor, remnant cancer cells scattered throughout the brain often regenerate into more aggressive tumors, something that just surgery can't fix. Chemotherapy is used to target those remnant cancer cells, but is still largely unsuccessful due to the presence of something called the blood-brain barrier. The blood-brain barrier is basically what it sounds like. It's a semi-permeable membrane that separates the blood from the brain. And the blood-brain barrier is essential for our survival. It allows for the passage of things we need like water and oxygen, while blocking out foreign substances like toxins and pathogens that might pose serious harm to our brains. However, this also means that it blocks out the anti-cancer drugs patients need to target those tumor cells within the brain. And this is largely due to the presence of a certain pair of proteins that are positioned along the blood-brain barrier, the P-glycoprotein, or the PGP, and the breast cancer resistance protein, BCRP. And here is a more in-depth look at the blood-brain barrier. Here are the two proteins, PGP and BCRP, and this right here is the glioblastoma, or the GBM tumor. And these little green things you see here are the anti-cancer drugs. So essentially, PGP and BCRP work together as a team of gatekeepers. Any anti-cancer drugs that try to pass through are promptly blocked and kicked out. However, there are three important facts to take note of. 1, Preliminary lab data has shown that PGP and BCRP are overexpressed in areas of the blood-brain barrier that harbor remnant cancer cells. Not only that, but GBM tumors have been discovered to secrete an inflammatory cytokine called tumor necrosis factor alpha, or TNF-alpha. And this TNF-alpha has been shown to signal upregulation of PGP and BCRP, or in other words, causes them to become even more selective on what goes in and out of the blood-brain barrier. So overall, we know that TNF-alpha has a hand in influencing PGP and BCRP to strengthen the blood-brain barrier against those anti-cancer drugs. But what we need to confirm is, what are the levels of TNF-alpha in patients with GBM tumors in comparison to those non-GBM bearing healthy patients? Or in context with this experiment, since I used mice, a more appropriate question would be, are TNF-alpha levels higher in GBM bearing mice than healthy control mice? And to test this, I used an enzyme-linked immunosorbent assay, or the ELISA. And what the ELISA does is it basically allows me to detect antigens or substances that can induce an immune response, like our TNF-alpha, among our assay antibodies. And for this particular experiment, I used two different types of ELISAs. First, I used the multi-analyte ELISA, which allowed me to detect the presence of a wide range of cytokines in my samples. And then I used the single analyte mouse ELISA, which allowed me to detect the specific quantity of a certain cytokine, the TNF-alpha, within my samples. And the procedures for both of the ELISAs were fairly similar. First, the wells of a microplate were coated with capture antibodies against certain cytokines. I then added samples and standards, and these cytokines, which are shown as these red dots within my samples, then bound to the capture antibodies that were pre-coded onto the microplate. Next, I added detection antibodies, which recognized and bound to the capture antibodies on the plate, which essentially created a capture antibody, cytokine, detection antibody sandwich. Avidin horseradish peroxidase was added to each of the wells, which then bound to the biotin within detection antibodies. And the combination of HRP and the development solution induced a color change reaction that was then halted by the stop solution. And the absorbance of the plate was finally read through a microplate reader in the lab. So this is my data for the multiplex ELISA, which again only showed whether certain cytokines were present within my samples rather than testing for these specific quantities. So graph G shows the data for the TNF-alpha cytokine, which was the highlight of my experiment. And because the levels of GBM are much higher than the levels of the negative control and the positive control, I was able to confirm that TNF-alpha indeed had a high presence within my samples. So after I confirmed the presence of TNF-alpha, I moved on to testing for these specific levels of TNF-alpha using the single analyte mouse ELISA. So before I go into the results of the ELISA, what I'd like to make a brief point is that the thing about GBM tumors is that they are not identical at all. All GBM tumors differ in slightly various ways. Or in other words, you'll never have two GBM patients with identical tumors. And for this reason, I used two different mouse models to depict my project. And each mouse model had a unique GBM tumor. For graph A, about 10 mice were injected with the U87 GBM tumor cell line. And for graph B, about 15 mice were injected with the U251 GBM tumor cell line. And the reason for this is so that if levels of TNF-alpha were shown to be high across those across a various range of tumors, I would be able to say that the results were actually significant, rather than saying that high levels of TNF-alpha were unique for only one GBM tumor. But back to the data, the GBM tumor mice were contrasted with non-GBM bearing mice that acted as a control. And if you look at the graphs, we can see that the levels of TNF-alpha are much higher than those of the mock control mice, whose levels of TNF-alpha were basically not detected, which doesn't mean that there wasn't any TNF-alpha within those mice at all. Something like that is actually impossible. We all have a little bit of TNF-alpha within ourselves. But going off the rails for a bit, but for example, if I had a cold, then that would mean that I have a lot more TNF-alpha within my system, because TNF-alpha is a cytokine that induces inflammation. So in essence, mock mice have a bit of TNF-alpha in them as well. It's just that their levels were too low to be detected by the microplate reader. But in conclusion, I was able to confirm that yes, GBM mice do indeed possess higher levels of TNF-alpha in comparison to the healthy control mice, which explains the signaling of upregulation of P2P and BCRP at the blood-brain barrier. But knowing about TNF-alpha isn't going to do much in the long run, since TNF-alpha is only a single variable. That's why my next steps include testing for the quantities of specific cytokines confirmed to be present within the multiplex ELISA, which would allow me to establish a pattern of biomarkers that I'd be able to recognize across patients with GBM, which hopefully would allow us to devise a treatment that would be able to target those specific cytokines within those patients. And I'd like to end this video with a few acknowledgments. Many thanks go out to my mentors, Dr. Bjorn Bauer and Dr. Annika Hartz, as well as everybody at the Bauer Hartz Lab for all of their support and guidance in helping me finish this research project. I went into their lab knowing absolutely nothing and came out knowing so many valuable lessons on what it means to be a scientist. I'd also like to thank the MSTC program and the Kentucky Young Researchers program for granting me this wonderful opportunity to dive into university level research as a high school student. I'd especially like to thank Mrs. Karen Young for all of her help and guidance and support during my time in the MSTC program. And finally, I'd like to thank my family and friends for all of their help with car rides, panic frenzies at science fairs, and for all of their support and love during my school years. Here is a brief look at my references. And for anybody with questions, feel free to contact me at my email. Thank you so much for watching, and I hope you had a great learning experience. Thank you.